The JAX Clinical Knowledge Base, also referred to as CKB, is a powerful digital resource that can be used to interpret complex cancer genomic profiles and identify potential therapies in clinical trials in the context of those profiles. We currently have 3 different offerings for CKB, as you can see from this slide. CKB Core, listed on the left here, is our free version that can be accessed online at ckb.jax.org. Within Core, users have access to 50 cancer-related genes and their associated content. The second offering is CKB Boost. Boost is another web-based version of the knowledge base, but requires a paid subscription. Users have access to all of the content within CKB, which includes more than 1,800 genes. Boost also includes advanced search capabilities as well as visuals. The last offering is CKB Flex. This is an option for users interested in downloading the content within CKB for integration potentially within a pipeline for tertiary analysis. A question we are often asked is, what does the content in CKB look like? Which is why we decided to create this video tutorial. Before demoing CKB Boost, let's take a look at the CKB data model. CKB, it's a relational database, so you can see from the data model there are multiple types of content that are linked to one another, including genes, and within those genes are gene variants, which are linked to molecular profiles. A molecular profile can include one or more variants. Molecular profiles are then linked to either efficacy evidence or clinical trials through indications and therapies. And therapies can consist of one or more drugs. Drugs can then be grouped into specific drug classes based on its target. Finally, connected to molecular profiles are treatment approaches, which can be assigned based on supporting evidence in the literature. Now that we've covered the data model, let's explore CKB Boost. Upon logging into CKB Boost, you arrive on the home page as seen here. If you've explored our free version, CKB Core, you can see it looks very similar, except that in this case you have access to all the search features in basic search as well as advanced search. So before we do an explore by gene, I'd first like to familiarize you with the tabs on the top right here. The first one is the news. So we post news items, and so here you can find both past and present news items. The second one is our gene grid. This gene grid includes all the genes that have been curated into CKB. So if you were to hover over one of these and click on the button, it'll bring you directly to the gene detail page. The next tab is the drug classes. So this is also shown as a grid. So within CKB, we assign drugs to drug classes, and the drug classes are named based on what they target. So for instance, ABL1 inhibitor, all the drugs, 6 drugs that is, within the ABL1 inhibitor drug class are targeting ABL1. And if you were to click on this, that'll bring you directly to the drug class detail page. The next one is the About. So here you'll find more information about CKB as well as the data model. The last tab here, which is called the Help section, if you click on this, it'll bring up a drop-down, and so you can see some information related to category variants, which I will go over, as well as the curation methodology. So we like to be very transparent with our curation in terms of explaining to users how we make decisions in terms of protein effect as well as response type for efficacy evidence. So the curation methodology includes some decision matrices. After that are the FAQs and then a glossary of terms. If there's any terminology within CKB that you're not familiar with, you can check here and see if you can find a definition. After that is the navigation legend, and this includes information related to the different types of buttons and the different colors and why they are colored the way they are, which is based on theme. And lastly, there's a tutorial, and this includes a PDF with screenshots of CKB. All right, so now we're going to do Explore by Gene. So before we do that, if you're ever within CKB and you want to get back to the home page, you just click on the top left here, and that'll bring you directly to the home page. Okay, so let's explore by gene. So the genes within CKB use HGNC approved nomenclature. So for explore by gene, you would click on this and then start typing in the gene symbol. I also want to point out that we do include synonyms for genes. So for instance, ERBB2, often referred to as HER2, you would start typing in the synonym and it should come up in the dropdown. We're going to explore the gene KIT. So I'm going to start typing it in, click on the kit, and hit submit. And this will bring you to the gene detail page. So here in the top left, you can see some information about the gene, its symbol, the synonyms, as well as the gene description. Our descriptions consist of 2 sentences. The first sentence describes the normal function or intrinsic function of the protein encoded by that gene. And the second sentence describes its association with cancer, such as maybe are there activating or inactivating mutations found or frequently found within a particular tumor type. And as you can see, everything here is referenced. So within CKB, if you see hyperlinks for PMIDs, you can link directly to PubMed, specifically to the paper, if you wanted more information. So content in CKB is referenced through literature that's indexed in PubMed, as well as through abstracts that have been published. So maybe abstracts from AACR or ASCO. Below that, you see some more information about the gene. So the NCBI gene ID, in this case, you can link directly to NCBI if you wanted more information. The chromosome and map location of the gene, the canonical transcript that we use for that gene, and then the gene role. The gene role could be an oncogene, tumor suppressor, both, or unknown, and this is the reference used to support that gene role. And then finally to the right here, you'll see some visuals. Now you don't see these in core. So the first one is a donut plot looking at protein effect of all the variants within KIT. So there are 293 variants. And so it kind of gives you the landscape of the different types of protein effects you'll see. And so when you hover over these, you can see the percentage as well as total number. The other tab is the variant impact donut plot. So again, in this case, you see the impact of those variants. So whether or not they're missense, deletion, duplication, and etc. So if you look below, you'll see 4 different tabs. The first tab lists all the gene variants. And as I mentioned, there are 293 for KIT. So looking at the table below, you'll see in the first column the list of variants, and these are at the protein level. The impact of those variants, so whether they're missense, nonsense, unknown, etc. The protein effect, so the controlled vocabulary within CKB includes gain of function predicted, gain of function predicted, no effect predicted, or unknown. And then the next column is the variant description. So the variant descriptions within CKB typically consist of 2 sentences. The first sentence describes the variant location within the protein, and a lot of this is taken from uniprot.org. And then the second sentence indicates whether or not the variant's been biochemically characterized. If it has, we'll describe the data for that characterization. If it is not actually characterized, we will indicate in that second sentence whether or not maybe it's been identified in a sequencing study, so a large spreadsheet where the variant's just listed, or perhaps maybe we might indicate it's been identified in the scientific literature, but they didn't actually characterize it. And so we'll include the PMIDs for these. With our variants of unknown significance, we also include the date, the last time that variant was searched on. And so within CKB, our SOP requires us to check every variant of unknown significance within 12 months. And then if you look over to the right here, you see a column called associated with drug resistance. So sometimes you'll see that certain variants may be described as resistant variants to a particular drug. And so in this case, we might include that information in the actual variant description. And so if I click the sort here and bring up all the variants that have a yes next to them, these are variants where you will see that information in the actual description. And this is specifically within the second sentence. And then of course the reference is there, so you can check if you wanted more information. This information will also be described in our efficacy evidence, which I will get to. And just to point out also that all of these tables, you have the ability to filter. So if I wanted only gain of function variants, I could start typing that in. I could also sort these columns. So if I wanted to sort them, I could do that and scroll down and see the percentage of variants that result in a gain of function versus predicted, and so on. All right, our second tab. These are category variants. So what are category variants? So we created category variants to capture information or variants that are not specific. So this might occur within FDA drug labels, the NCCN guidelines, or even clinical trials and publications. And so oftentimes they don't specify the actual variant. So it might say EGFR activating mutations. And when it's a study, we don't necessarily want to guess what those variants are. We want to stay true to what the literature is saying. So we created two types of categories. They could be functional or positional. Functional would be your ACT mute, and then positional would be your variants that occur at a particular exon or maybe at a particular codon, and the amino acid could be any change. And so with category variants, they can actually be used as parental terms for specific variants. And when I do an explore by variant, you'll see how that plays out. The next tab over is our molecular profiles. So we created the concept of molecular profiles in CKB in order to capture times where there might be more than one variant associated with evidence. So often you'll see that tumor samples may contain more than one variant and be related to the response. So in this case, we can create what we call complex molecular profiles, or maybe single molecular profiles where there's only one variant. And so if you look below, you can also see some treatment approaches in this last column. So if I were to sort this, you can see variants in which there's been certain drug classes assigned to them, and that may be indicative of an actual treatment approach. The last tab, which is considered to be the bread and butter of CKB, this is our efficacy evidence annotations. So an efficacy evidence annotation links a molecular profile, which can, like I said, can consist of one or more variants, to a tumor type, a therapy, and a response, okay? So if we look at the table below here, we can see that we have 573 different efficacy evidence annotations. And so for every efficacy evidence line, there's a particular molecular profile. And that profile, because we're on the KIT gene detail page, must contain a KIT variant. However, it could contain variants from other genes as well. And so in the first column is your actual molecular profile. In the second column is the tumor type. Now we use the disease ontology to assign tumor types. The column after that is the response type. So within CKB, we have controlled vocabulary for response type, and that could be sensitive, resistant, predicted sensitive, predicted resistant. It could be not predictive, conflicting, not applicable, or could also be unknown. And finally, decreased response. The column after that is the therapy name. So therapies, we can basically build therapies. So therapies can consist of one or more drugs. So what you'll see is the drug that was used in that particular study. The column after that is the approval status. And this is where you'll see your high-level evidence versus lower-level evidence. So at the high level, you've got your FDA approvals, guidelines, and then as you scroll below that, you'll move down to clinical trial data, so from phase 3 to phase 1, and then clinical study data if it's not specifically a trial, then case reports, and then finally preclinical data. And the way that we group our preclinical data, it could be separated out based on cell culture, patient-derived xenograft models, cell line xenograft models, or patient-derived cells. Now, if we scroll back up, we see the next column is evidence type. So this describes the different types of evidence within CKB. So the majority of the evidence in CKB is actionable. However, we do include some evidence related to diagnostic, prognostic, risk factor, and then there might be some emerging evidence as well. The next column is the actual efficacy evidence annotation. So this is a free text annotation. So it consists of one sentence, and it's basically a brief synopsis of the results of the study. So for instance, if we look at this first one, What we do for FDA drug approvals is we actually go to the label, figure out what is the study that was used to support that FDA approval, find the publication, and then write the evidence annotation. And as you can see, everything is referenced. So within the clinical data, if it's a trial, we will include the primary endpoint results as well as the secondary endpoint results. And again, you can always reference out to PubMed if you need to. And so this is basically what you will see for pretty much every efficacy evidence annotation. They're all going to be one sentence. Okay, so let's look at these last two columns here, evidence level and inferred tier. So we utilize the AMP-CAP ASCO evidence levels and assign them to the efficacy evidence. So each annotation is going to have its own evidence level. So anything at your high level evidence, such as FDA approvals, is going to be evidence level A, and then obviously decrease as you get to the preclinical. We also include the inferred tier for the variant related to that efficacy evidence. And we say inferred because it could be possible that the sample you have might be a different tumor type. And in that case, the tier would be downgraded. So now that we've looked at the gene level evidence within a gene, the variants, the category variants, and the molecular profiles, let's move on to explore by variant. Say there's a particular variant you have in a patient sample, and you want to know what's the protein effect of that variant. You would do this by doing explore by variant. So you'd click on this, and you would start typing in your variant. Now, just to point out, the variants are curated using HGBS compliant nomenclature. So you'd start typing in your variant, and we're going to go along with the theme of KIT. So let's type in KIT K558 underscore Y570 del insertion R. Okay. If the variant is in CKB, it will come up in the dropdown. If it's not in CKB, you can always request curation. We welcome people to email us at ckbsupport@jax.org and send us variants, and we will curate them into the database. So once your variant pops up, click on it, hit submit, and this will bring you to the molecular profile detail page for that variant. So we're not going to go over the molecular profile detail page in this tutorial. Instead, I want to bring you to the gene variant detail page. So here at the top, you see the actual profile name and then the actual gene variant detail. If I hover over this, you can see the variant description. But let's click on it. That brings us to the gene variant detail page. In the top left, you can see the information related to the variant. So the gene, the actual variant, the impact, the protein effect, and then the variant description. And so this variant, you can see just based on the description that it's predicted to result in a gain of function. And this is based on other KIT exon 11 deletions that are activating. And these are the references to support that. Below that is where you see those category variant paths. So when we did explore by gene, I talked about category variants. So when you have specific variants, such as this one, you can actually add them as a member to a parent category. So in this case, KIT exon 11 deletion, it acts as a parent to the specific KIT variant that we searched on. And then KIT exon 11 deletion can then fall into KIT exon 11, and then KIT exon 11 into KIT mutant. And then the same goes for this second path, except this one, it falls into ACT mute. So I mentioned there are two types of category variant paths that can be presented, either a functional path or a positional path. So the KIT exon 11 would be your positional, and the KIT ACT mute would be your functional one. In the top right here, you have the variant reference transcript. So you can see information related to the actual transcript, the gDNA, cDNA, protein, etc. And if you had other transcripts you were looking at, you could check this tab as well. So what I want to point out to you is the evidence below here in these different tabs and how the category of variant paths can help with identifying potential treatments based on the variant that you searched on. So if we look at this first tab, we see evidence, and this would include efficacy evidence directly related to the variant that you searched on. In this case, you see there is no evidence for that variant. However, if we click on the second tab, which indicates extended evidence. This includes efficacy evidence that is specific to these category variants within these two paths. So looking at the table below, it looks exactly the same as the table you would see on the gene detail page in terms of the columns. And you can see at the top, this is your high-level evidence. And so the different category variants that you see in the paths are going to be listed here. So for instance, if your patient has melanoma and has this particular KIT mutation, it's going to fall under ACT-MUT. And so you can see that this efficacy evidence, which is actually associated with a guideline, that the drug imatinib is included in guidelines as second-line therapy for melanoma patients with KIT activating mutations. So this could be one potential therapy in that case. And so as you scroll down, you can see there's other types of evidence. And of course, as you get down below, that's going to be your lower level evidence. So this is just one way in which you can identify a potential treatment for a patient. Okay, so we've done explore by gene and explore by variant. I'm going to go back to the home page. However, we will be doing other video tutorials, so please stay tuned for explore by variant, specifically looking at molecular profile detail page, as well as drug class, drug, tumor type, and then the advanced searches. At this time, though, I thank you for watching our video and exploring CKB Boost with me.